Hello, and welcome to part two of your biology portfolio projects. So let's just jump into it. In this section, we're going to be taking a look at a project dealing with DNA and mutations. And this one you're going to be able to make out of, well, I'm going to give you the option to make it out of candy because why not? Why not be able to make something and be able to admire it and then afterwards be able to eat it? That's my philosophy. So without any further ado, let's take a look at what exactly it is that you're going to be expected to do in this particular section. As you can see here, we're going to be taking a look at the mechanisms of genetics. These are the expectations. You need to complete all of the following. Use candy or craft items. As I said, I'm going to give you the option of making it out of candy because it's fun. To create a model of DNA, you can use Twizzlers for the frames, Skittles for the nucleotides, or you can come up with your own way, gummy bears, gumdrops, milk duds, I don't care. Just be creative when you come up with how you create your own model. Make sure to label which candy or item represents which part of the DNA strand. Be sure to label the 4 base nucleotide pairs, A, T, C, and G. Also label the deoxyribose backbone of the model, or the sugars that everything connects to. Also include examples of the correct pairing, and include and label 3 sections that show an example of a mutation, such as a substitution mutation, a deletion mutation, a transposing mutation, or any other kind of mutation that you wish to show. Once completed, you will take pictures of it and you will email them both, or you will share those pictures with myself and Miss Cowan. Now let's take a look at the grading rubric, shall we? In this particular rubric, you're going to create a model of DNA using recycled or found materials. Use this as a guideline to make sure that your project is done correctly. You're going to have at least a 20 base pairs minimum. In other words, 20 connections between AT, GC, GC, CG, TA, whatever, 20 of those at the very least. I would highly suggest doing 4 sections of 5. Have the very first section, whether it's the top or the bottom, be the correct section where each one actually matches up and is correct in their pairing. Then the next section of 5, you can actually repeat the correct section, but then or the correct section pairs, but then actually include an example of some kind of mutation, whether it's deletion, whether it's duplication, whether it's transposition, whether it's substitution, whichever one that it actually is, that's what you need to do, and then label it as such. And then go to the next section of 5, and then show another example of a mutation. And then go to the last section of 5 and show another example of a mutation. And then as you do, you can label each section as the correct section, such and such mutation, such and such mutation, and so on and so forth. And so in that way, you would have 4 sections of 5 base pairs. One section is the correct section. Be sure to label that. The next section has a mutation. The second section has another mutation, and the third section, or the fourth section, has a third mutation. That would cover all of your bases for the 20 base minimum pairs, showing the fact that the nucleotides are bonded properly in the correct pairs, and then the examples of the 3 mutations. Now, these are the examples of mutations. They do not have to be these exact mutations, but they do have to be a mutation. So here are the mutations, and so you'll get points of for each mutation that you show. And then also the points, you'll also get 4 points for the fact that all of the base pairs connect to the deoxyribose sugar ladder, as it were. And then that if it's shaped into the correct double helix shape. The quality of the model, make sure that the DNA model is made of the proper materials that we've talked about. Make sure that you include a key or labels. If you include a key, make sure that you know that I know that every red gummy bear or red Skittle or red Starburst or anything such as that equals an A or equals T or whatever, and that Twizzlers or rope or pipe cleaners or whatever it is that you use for the deoxyribose, that is the deoxyribose sugars. And then, like I said, make sure that you label each section as the correct section, as whatever particular mutation. If you're going to use duplication, make sure that you include the duplication mutation there, the substitution mutation, and then the deletion mutation, whatever it happens to be. Make sure that you just label each part correctly. Make sure that it is excellent and neat. In other words, make sure that it's not a mess and that I can clearly see what it's supposed to be. I can clearly see what it is that you're trying to do. If I can do that, then awesome. Make sure that it shows creativity and time invested, not something that you just slap together in 5 minutes. Make sure that I can tell that you spent some time on it. Label everything is going to be one of the key points on this. Make sure that if you can print it out, print it out. If it's handwritten, make sure that it's written in the nicest handwriting possible. Make sure that it is neat, and then you're going to receive an extra 5 bonus points if you can take a picture and it actually is freestanding. In other words, it can stand up on its own, and it has nothing touching it, nothing propping it up. It's just standing from the base, and you're able to just set it on the ground and it just stay there. So if you can do that, kudos to you, and you get an additional 5 bonus points. Now here's where the points fall. For an A, you need to get anywhere from 56 to 62 points, because that's as many of those added up. If you want to receive a B, then it's 50 to 55 points. A C, 43 to 49. D, 37 to 48. And then, of course, an F is 36 points or below. So if you have any questions on that, by all means, please feel free to contact me. Now, this can be a very easy thing to receive directions on, because all you have to do is Google DNA models from candy, and the very first thing it pulls up is how to make a DNA model out of candy. And so if you click on this, it will show you very easily and very carefully, and it will give you step-by-step instructions on how to indeed, or at least give you some examples on how to indeed create DNA models out of candy. It even gives you suggestions for the materials. It gives you suggestions for what to do on that. But remember, not only do you need to create it, but you also need to label it, and you need to show examples of mutations. So this is only going to show you how to do the correct ones. Make sure that you include the mutations in there as well. Anything else, please feel free to contact me with any questions. Once again, I know that you are going to do amazing. I know that this is going to be fun, or at least it can be if you let it. So once again, thank you and have a good day.